a new microarray designed to use as a universal cancer diagnostic tool for detection of fusion genes. Long and tough title. I will use the 12 minutes to try to explain what this is. And also this is something that we hope will be a molecular diagnostic tool also for colorectal cancer in not too long. So, a fusion gene, that is something that is created in very many cancer cells. These are two chromosomes, red and blue. Apart from the red chromosome can switch part with the part from the blue chromosome, and if this happens in the middle of two genes, which very often happen in cancer cells, for example inside what is called an intron, parts within a gene that is not coding to a protein, then a fusion gene is made when the resulting fusion gene is actually having parts of whole coding exons from one gene and whole coding exons from the other gene. And this is caused by chromosomal translocations, deletions, or inversions in the tumor genome. And cancer cells have many of these changes, and it's particularly common in hematological cancers, which is leukemias and lymphomas. It's also very common in sarcomas and in prostate cancer. And it's because of poor ways to detect these fusion genes that we also don't know many such fusion genes for cancer types like colorectal cancer, breast cancer, and they are definitely common also in more cancer types. So we need, and the thing is that identification of certain fusion genes, these things are currently performed for differential diagnosis and therapeutic decision making in hematological cancers and sarcomas. And the way people are doing this in routine diagnostic laboratories is mainly they are looking at the chromosomes under a microscope, or at least imaged under a microscope. This is a very inefficient methodology because you have to grow the cells to make these chromosomes look like this, and that's only possible in about 70% of the cases. And it's also very low resolution, so you don't see many of these fusion genes by this technology. And then there are more specific technologies like PCR-based and fluorescence in situ hybridization. They are when you have a hunch for what you're looking for, what kind of fusion gene you're looking for, they are precise. But there are about 500 known fusion genes. And if you don't know what you're looking for, it's too much to do a PCR reaction from 500 tests. And also, you don't know, if you have this particular fusion gene, then you don't know whether it's this blue exon you should have joined to this red exon, or whether it goes from this exon to some other red exon, and so you have to make many more than the 500 PCR tests if that's the method you're going to use. And this is the case for prostate cancer. It was only 3 years ago that the first fusion gene was detected in prostate cancer, and already today it's now fusion genes from 10 different upstream gene partners to 5 different downstream gene partners. And again, this is very much simplified because the fusion can also go from the other exons in both the upstream and in the downstream gene. So to test for this with a PCR-based test, you would need hundreds or even thousands of tests only even for the prostate-specific fusion genes, which are relatively few. So, what is our solution to this? We are, if you have a fusion gene like this, and the fusion that goes from this exon in the one gene to the other, we make a oligonucleotide probe targeting exactly the exon-to-exon junction after the intron is spliced out. Because almost all the fusion breakpoints they have inside the intron. And that makes it possible to detect with the same oligo detector the same fusion, whether it happens here or it happens there inside of the intron. And by multiplexing all of this, so we go from all the exons in the upstream gene to all the exons in the downstream gene, and also by doing this for all the known fusion genes at the same time, it's possible to detect all potential fusion genes in one single test. When we did the pilot design, we knew about 275 of these, and by retrieving sequences from genome databases and generating all the chimeric sequences, which were about 60,000 for the pilot design. We made a microarray that we made into a single glass slide, the size something like this, with all the oligo sequences synthesized onto the slide. And we used the Nimbogen platform to do that, which is a technology that allows us to send a text file with anything up to 2.1 million sequences to the Nimbogen company, which at the time of the pilot was placed on Iceland. And then sending them the text file with the sequences, they send us the slides back, and we can use this in the fusion gene detection tool. In the pilot study, we studied 6 different samples. Of course, when we have tens or hundreds of thousands of measurements, we need some kind of automated scoring. I will not go into the algorithm of this, but it's showing the results for one of the prostate cancers here, picking up the 2 duplicates from the fusion gene as the top scoring hits among some tens of thousands of possibilities. The pilot was recently published in Molecular Cancer a couple of months ago,